Liger. Lions and tigers split from each other roughly 3.7 million years ago. Their genomes were never meant to recombine. When they do, what you get is the largest cat on Earth. Males regularly exceed 900 pounds, dwarfing both parent species by a significant margin. But that size isn't a feature, it's a malfunction. Both parent species carry growth-inhibiting genes that suppress size after a certain developmental point. The liger inherits neither set correctly, so it simply never stops growing. The result is an animal evolution never designed and biology never intended to sustain long-term. Male ligers are always sterile because of mismatched sex chromosomes. Most suffer cardiac problems, neurological issues, and dramatically shortened lifespans compared to either parent. The size looks impressive until you understand what's actually happening underneath it. Two fundamentally incompatible genetic systems colliding inside a single body, and the body absorbing that collision for its entire life. Grolar bear. Polar bears and grizzlies split from a common ancestor somewhere around 500,000 years ago, and spent hundreds of thousands of years adapting to completely separate environments. Polar bears evolved for sea ice, seal hunting, and brutal Arctic conditions. Grizzlies stayed on land, omnivorous, territorial, built for temperate forests and mountain terrain. Their ranges didn't overlap. They had no reason to meet. Then, the ice started disappearing. The first confirmed wild grolar bear was shot by a hunter in the Canadian Arctic in 2006. DNA testing confirmed it. Since then, second-generation hybrids have been documented, meaning these animals are fertile and actively backcrossing with parent species in the wild. That's not a curiosity. That's a population-level genetic shift happening in real time. Grolar bears inherit the polar bear's fearlessness toward humans combined with the grizzly's territorial aggression. Climate change didn't just force two species into the same territory. It's actively dissolving a genetic boundary that held for 500,000 years. And it's doing it within a single human lifetime. Wolphin. False killer whales and bottlenose dolphins belong to entirely different genera and are separated by millions of years of evolutionary divergence. They share no meaningful overlap in social structure, prey, or body size. A male false killer whale reaches up to 20 feet in length. A bottlenose dolphin tops out around 12. The size gap alone should make viable offspring nearly impossible. And yet, there are two wolphins alive right now. The first, Kekaimalu, was born at Sea Life Park in Hawaii in 1985 after a false killer whale and a bottlenose dolphin were housed together in captivity. She survived, and then she reproduced, giving birth to a calf of her own, which makes wolphins not just viable hybrids, but fertile ones. Fertile hybrids across genus lines are extraordinarily rare. Most intergenus crosses produce animals that can't reproduce at all. Scientists still don't fully understand why the genetic barrier between these two species is thin enough to allow it. There's no clean answer. The wolphin exists as living proof that evolutionary separation and genetic separation are not always the same thing. Zorse. Horses carry 64 chromosomes. Zebras range from 32 to 46 depending on the species. Cross them and the offspring inherits a mismatched chromosome count that makes meiosis, the cellular process that produces sex cells, functionally impossible to complete correctly. Zorses are always sterile, without exception. That's the chromosomal problem. But the behavioral incompatibility runs just as deep. Horses have been selectively bred for thousands of years to tolerate human handling, follow commands, and work alongside people. Zebras have never been domesticated. They've been bred by nothing except survival pressure in one of the most predator-dense environments on Earth. Zorses inherit the zebra's aggression, its skittishness, and its flat refusal to cooperate under most circumstances. The original goal was practical: a disease-resistant pack animal capable of surviving in tsetse fly regions of Africa where horses couldn't. What was produced instead was a sterile animal that chromosomally cannot reproduce and behaviorally will not be managed. Every layer of the biology said this wouldn't work. The behavior is just the most visible conformation. By the way, if you're enjoying this, I post content like this regularly. I'd appreciate if you subscribed. Thanks. Coywolf. This one didn't happen in a lab and nobody planned it. Eastern coyotes across the northeastern United States and southeastern Canada have been DNA tested, and the results consistently show wolf and domestic dog genetics woven directly into their genome alongside coyote DNA. It happened in the wild, as wolves were systematically eliminated from eastern North America through the 18th and 19th centuries. Coyotes expanded into that vacated territory and encountered the last remnant wolf populations and feral dogs along the way. Coyotes, wolves, and dogs all belong to the genus Canis, so the chromosomal barrier between them is low enough to allow fully fertile offspring. What emerged from those crossings is larger than a pure coyote, with a more wolf-like skull structure, significantly stronger jaws, and the behavioral capacity to hunt in coordinated packs, something coyotes don't typically do. It also carries reduced fear of humans inherited from the dog genetics, making it exceptionally well adapted to urban and suburban environments where pure wolves would never survive. The coy wolf now dominates the Northeast. It isn't a dead end. It's actively outcompeting both parent species in human-altered landscapes. Habitat destruction and predator elimination forced 3 separate canids into contact. The hybrid that resulted is winning. Blood parrot cichlid. This fish makes the genetic incompatibility physically visible in its own body. The blood parrot cichlid was created by crossing 2 Central American cichlid species, most likely the Midas cichlid and the redhead cichlid, by commercial breeders in Taiwan during the 1980s. The exact parent combination was never publicly disclosed. What is documented is the result. The 2 parent species carry incompatible skeletal development pathways, and when those pathways conflict during development, the offspring pays the price. Blood parrots have mouths that cannot fully close, making normal feeding mechanics impossible. Their spines are compressed in ways that crowd internal organs. Swim bladder dysfunction is common, affecting their ability to control buoyancy. Males are sterile in most cases. These aren't minor or incidental defects. They are structural failures baked into every individual of this species from the first cell division. The fish is bred commercially by the thousands and sold globally as an ornamental pet because the orange-red coloration and compressed heart-shaped body profile test well in the aquarium market. The genetic incompatibility is not an unfortunate side effect of the breeding program. It is the product. A dromedary camel weighs approximately 1,400 pounds. A female llama weighs around 300. The size difference makes natural breeding physically impossible. The only way to combine them is artificial insemination, which is exactly what researchers at the Camel Reproduction Center in Dubai did in 1998. The stated goal was practical: combine the llama's fine wool with the camel's size and carrying capacity to create a more useful pack animal. What was produced was a small, llama-proportioned animal with camel features including short ears, a long tail, and no hump. Temperament was described by the research team as unpredictably aggressive and difficult to handle. Camas are sterile. Camels and llamas are separated by approximately 30 million years of independent evolution on separate continents. Camels in Asia and the Middle East. Llamas in South America. They never shared a habitat. Artificial insemination made the size barrier irrelevant and temporarily overrode 30 million years of divergence. The biology disagreed immediately. The project never expanded beyond a small number of experimental animals. Narluga. A skull sat unidentified in the Natural History Museum of Denmark for decades. An Inuit hunter had shot an unusual whale in Greenland in the 1980s and kept the skull, which was eventually donated to the museum. The teeth didn't match any known species, intermediate in structure between a narwhal's single spiral tusk and a beluga's small conical teeth. In 2019, DNA analysis confirmed it was the offspring of a female narwhal and a male beluga. Narwhals and belugas are distinct Arctic whale species with different prey, different dive profiles, and different vocalizations. They share overlapping Arctic ranges, but were assumed to maintain reproductive isolation. That assumption was wrong. The confirmed specimen is one animal. Whether more narlugas have existed, born, lived, and died without anyone testing the DNA, is entirely unknown. Both species are are under compounding pressure from sea ice loss and shifting Arctic conditions. If their ranges continue to overlap with increasing frequency, more narlugas may already be out there. The skull sat in a museum drawer for 30 years before anyone knew what it was. That's 8 animals biology said couldn't exist. Some are sterile, some are fertile and spreading. One is actively reshaping an ecosystem, and at least one more might already be swimming in the Arctic, undetected, waiting for someone to run the test. If this sparked your curiosity, consider subscribing so you don't miss future content. Thanks for watching.